All right. So you should be able to see my screen, and if that's a problem, someone let me know. But so today, like I said, I'm going to sort of talk to you a little bit about the work I've been doing studying how animals behave and respond to climate change and their rapidly modifying environments. And that's sort of the general research questions that make me curious at large. Are animals and the sort of unique ways that they're coping to a human dominated world. Um, and so, how do animals cope with rapid environmental change? Um, and that becomes a more and more relevant question in our current state of affairs, especially when we think about climate change. So climate change is rapidly modifying habitat, and many organisms are shifting their ranges either to higher latitudes or higher elevations in response. However, these range shifts aren't completely identical across species and across locations and contexts, and they can often be quite heterogeneous, and similar species can have vastly different responses. And understanding this variation is going to be critical for our approach, for how we approach conservation and management in the future. My research in particular is really concerned with high altitude species. This is just one study out of many that's reporting the commonly observed phenomena that high elevation, high altitude species seem to be much more sensitive to changes in climate. This quote here from a study in the Australian Alps showing that among alpine endemics, so species whose range is entirely at high elevation, um, they're, they're poised to experience a 77% loss of habitat even under the weakest climate change scenario. And so in a lot of ways, alpine species are at the front lines of climate change and climate adaptation as our planet continues to warm. And this is an issue that's particularly dear to the hearts of those of us who work and live in the Eastern Sierra. Many of the animal species who we coexist with are only found at these high elevations. And they're like the animals all over the world are at particular risk from a changing climate. These are just a few of my favorite species that live up in the High Sierra, other than the chipmunks, of course. This will be your bird content for the day. Sorry about that. You know, this could be a man talk here. And so we're starting to understand that climate is particularly has a particularly strong effect on species living at high elevation. But how do we know that? How are we determining that species are being affected by climate? How do we know their populations are growing or shrinking, or their ranges are growing or shrinking? And one of the ways we do that is with museum data. And so at the University of California, Berkeley, I'm based in the Museum of Vertebrate Zoology, which studies living animal diversity with particular focus in California, but from animals all over the world. And this museum has been in operation for about 100 years or so. And the original museum's founder, Joseph Cornell, was very ahead of his time in how he approached museum science, and that not only were museums a place to sort of collect and house and archive physical material, but the data and the information that came with that material was just as, if not more important. And so when Joseph Cornell and 100 years ago and the other museum staff were doing their collections, they took meticulous notes of the metadata, location information, what the animal and plant communities were like, what the conditions were like at the collection site, and archive that in their field notes. And so with that rich resource of archival information, museum scientists 100 years later can go back to that material, see their notes, and then redo those same surveys. And so you have surveys done 100 years ago and surveys done in the present, and if you follow their specific instructions and procedures very carefully, you can get a really good idea of how different locations across the state, how their animal communities have changed over time. And that's exactly what the museum did with the Grinnell Resurvey Project, which was funded by the NSF starting about 15 years ago now, and it's still ongoing. But a lot of the work I'm talking to you about now is is work done in the mountains, which was sort of the first leg of the Grinnell Resurvey project. Excuse me, a little sniffly. But basically, to accomplish this project, the museum sent out extensive field teams to sample all across the Sierras and Lassen in Northern California, all the way down into Southern California and the deserts as well, sampling the sites that Grinnell sampled 100 years ago with similar techniques, similar effort, to then get an accounting of the animals that live there, and then compare that to the past surveys to get a sense of how the populations have changed over time. And so we don't have, and so this figure's a little busy, and don't get too caught up in it. But basically, this is all this is to show you is a result from one of those papers looking at mammals of the Sierras across that 100-year time period. And as you can see, this color-coded figure, we have yellow bars indicating range expansions, red bars indicating range contractions, and then gray and white indicating non-significant expansions and contractions respectively. And what this shows you is that while there's sort of a pattern here in that in general expansions are happening at the high elevation edge of the species range, and contractions tend to happen at the lower edge of the species range, which lines up with our understanding of how high elevation species are responding to climate change. This isn't a 100% pattern. There's a lot of variation in which species are either expanding upward or contracting upward, and some species seem to be have very little change in their ranges at all. And so to zoom in a little bit, the species that I'm interested in are two of our chipmunk species that call the Eastern Sierra home. One of those being Tamias speciosus, the lodgepole chipmunk, and Tamias alpinus, the alpine chipmunk. And so these two figures on the right here are sort of a zoomed in version of that really big figure, showing what capture localities for each of these species based on elevation. And what we can see with the lodgepole chipmunk, Tamias speciosus, is that while there's a few lower elevation sites that no longer have the lodgepole, on the whole, it still occupies the vast majority of its historical range. We have very little change in its elevational range over this 100-year time period. Meanwhile, Tamias alpinus, the alpine chipmunk, we see really large upward contractions of its lower elevational range in both southern and northern sample localities. And that really raises some interesting questions, because these two species, anyone who's hiked up above 9,000 to 10,000 feet, not that I expect the average person to be able to identify tiny chipmunks to species like I can. I barely can. But if you notice, if you have seen a lot of chipmunks and know you can tell the difference between the two, you'll often see that these two species co-occur really close to each other. They're neighbors. And often when I'm doing my trapping, I'll set trap grids and I'll catch both individuals along the same grids within meters of each other. And so they occupy similar habitat. They live in similar environments. They tend to be eating somewhat similar things, or have at least similar availability of resources. Yet we see this really big difference in that change in elevational range over time. And so that raises a really good question. What's different about these two fairly closely related species, where one seems to be resilient to climate change and one seems to be really sensitive to climate change? And so I'm not the first to look into sort of what structures the ranges of our chipmunks in the Eastern Sierras. Some really wonderful work by a group of scientists in the 70s looked at all four of the species of chipmunks that inhabit sort of the eastern slope of the Sierras. So we have Tamias speciosus and alpinus, who are kind of more on the top higher elevational range. We have Tamias aminus, the yellow pine chipmunk, which is a mid-range species. And then we have Tamias minimus, which is our kind of lower elevation basin species. That's the one you're going to see running around the sagebrush down by the lake. And so what we see here with these four species is they seem to partition themselves very neatly by elevation. Again, with minimus at the bottom, then the yellow pine chipmunk amenus, then speciosus, the lodgepole, then alpinus, the alpine. And what we're thinking, and what was the thinking then is that individual physiological preference, so the actual physiological systems of the animal play into these habitat preferences on the landscape. And so we're thinking that behavior and physiology, so how the animal's body condition as well as how it interacts with its environment and with other animals in its environment, help to structure this. And then not only are they reacting to the environment and spacing themselves based on the environment, but then these boundaries are maintained by strict species interactions at the range edges as well. And so in these studies by Chappell, experimental increases of body temperature reveal that these different species have different temperature preferences. So low elevation minimus species can remain active at much higher temperatures than any of the other species, and can live in a much wider variety of habitats, while these other more temperature limited species are more restricted to the much patchier forest habitats. And so these more specialist species exclude their competitors from highly productive habitat, while these more subordinate species adapt to the more marginal habitat. And so we kind of see, and so that's kind of what we see at the higher end. So minimus being at the lower end, my two species up at the higher elevational end. So Tamias speciosus and Tamias alpinus. Stable isotope data, changes in skull shape, and loss of genetic diversity all tell us that Tamias alpinus, the alpine chipmunk, is more of a specialist, moving towards a specialist ecology in response to this past 100 years of climate change. How do we know this? But how do we know this for an individual? So we have information at the population level about genetics and trends in skull shape, but do we see this reflected in individual members of the species? One of the easiest ways, or one of the best ways we have to start to untangle that is looking at the physiological stress response. And so if our hypothesis is that the environment is at some level non-advantageous for this individual, this should probably reflect in its stress response, in its physiological stress response. And so these physiological stress response provides immediately mechanisms for organisms to respond to their environment. And we measure these stress responses through glucocorticoid hormones, or GCs, as they're called. And so they're important indicators of short-term responses to environmental changes. The scary-looking diagram on the right just shows the neuroendocrine pathways that lead to the manufacture of this hormone. And so this is on the hypothalamic adrenal axis. And so this neuroendocrine pathway is associated with metabolism and with immune response and with stress. Stress, of course, is a really big and nebulous umbrella term that indicates a lot of different physiological aspects of an organism. But here we're talking about stress in the broad sense of when you're responding to suboptimal environmental conditions and your body has to respond to compensate for that. And so one of the best ways we have to measure baseline stress, or think about what stress an individual is dealing with on a general level, is to measure stress in the feces, so fecal glucocorticoid metabolites, FGMs. And those have a lot of advantages over taking GC measurements from, say, blood, because sort of the GC levels that are circulating in the blood represent kind of an instantaneous snapshot of the stress levels. And you can imagine you're handling an animal that's there's one thing that's going to be stressed about, and it's going to be you. So those fecal glucocorticoids give us a more representative time series of what the stress they're dealing with from a day-to-day basis. And so for the animals, when we want to measure fecal glucocorticoids, we collect feces from the animals. And so when we're doing our normal captures, we'll collect feces and then use enzyme immunoassays to isolate and then measure concentrations of those fecal glucocorticoids. Previous members of my lab done experiments validating these methods and using experimental glucocorticoid assays to stimulate stress responses in the lab and then measure the glucocorticoid response. And so this is kind of a graph from here, the blue line representing where a glucocorticoid stimulating hormone was injected into an animal, and then the sort of the time delay of 24 hours were the peak. And so you can see the top is Taenius alpinus, the bottom is Taenius speciosus. And we can already see, even in this very neutral experimental scenario, that alpinus species, not only do they have this peak in stress, but they take a much longer time to return to baseline than Taenius speciosus, which is really fascinating because, again, these aren't even individuals. These are individuals who are being held in captivity temporarily. And even in captivity, we're seeing this vast difference in how they react and attenuate to stress. And so that gives us further evidence that even on an individual level, Taenius alpinus seems to be a lot more sensitive to fluctuations and changes in its environment than Taenius speciosus. What about on the landscape scale? And so on the right here, we have two graphs looking at various sites with different average daily temperatures and measuring kind of average FGM levels for each of the species. And in Taenius alpinus, we see a positive relationship between stress and fecal glucocorticoids or stress hormones, a relationship we do not see in Taenius speciosus. And then not only do we see that relationship, we also see a lot less individual variation in Taenius alpinus and Taenius speciosus, which is also fascinating. And so what that tells us then is that Taenius alpinus exhibits greater response to acute stressors than Taenius speciosus. Taenius speciosus exhibits greater individual variation in physiological stress responses. And so the question is, okay, we know how these stress responses are starting to differ between these species. What does that tell us about this population level response that we're seeing? And what do you do to respond to those environmental stressors? Well, you got to be behaviorally flexible, right? And so that's kind of where I come in. So a lot of the work, the physiology work, was done by folks in the past in my lab. And so my approach then is to see, okay, we know that they're stressed. We know that there's variation in environmental responses. Do they behave differently due to those environmental stressors and change? And so two of the behaviors that I'm interested in are exploratory behavior and boldness. Exploratory behavior is an animal's tendency to explore and sample novel environments. And boldness is an animal's willingness to engage with a novel, discrete stimulus. So these are not necessarily, these behaviors are quite related and often are intertwined. But you can imagine when you're in a changing environment, when you're dealing with novel and unfamiliar habitat conditions, being able to explore and sample and learn new things about your environment can be a really potent survival tool to increase your toolkit and access to resources and help you survive even when the environment is becoming more and more unpredictable. So we have our two species here. We have Taenius speciosus with its wide geographic range, a generalist ecological niche, and a decreased stress response. And then we have Taenius alpinus with its small geographic range, a specialist ecological niche, and an elevated stress response. And so what do we think that's going to affect their behavior? Well, I would predict that our speciosus, our lodgepole chipmunk, is going to be much more bolder and much more exploratory. It doesn't respond, it doesn't have as much of a physiological stress response. Its population has been able to withstand a century of climate change, which leads me to believe that that individuals of the species are more willing to explore their environment, engage with novel and unfamiliar resources and habitats, and are better equipped to survive. While our Taenius alpinus individuals, with their specialist ecology, smaller range, and elevated stress responses, are much less bold and exploratory. Those elevated stress responses shows that their physiology is more acutely tuned to deviations from the normal. And so over the past 100 years, in response to this shifting environment, it seems that the alpine chipmunks have been doubling down on the strategy that's worked for them for millennia, becoming more specialized. Because while the lodgepole chipmunks can exploit a wider variety of resources, the alpines are much more effective at exploiting the resources that they're specialized for. And so the big picture question is Alpine generalis Tamias speciosus, the lodgepole, more behaviorally flexible in response to a changing environment than the Alpine specialist Tamias alpinus? And then I predict that Tamias speciosus individuals will tend to be more bold and more exploratory, as well as exhibiting greater individual variation than Tamias alpinus, the Alpine chipmunk. So now I've got the questions, I've got the hypotheses. Now I have to actually go and study this and figure this out. And so I do most of my work right here on the east side, right up the mountain from Mammoth. A few of my sites are kind of along Tioga Pass, Highway 120, particularly around Saddlebag Lake and the Hoover Wilderness, and also I have some sites further south than Mammoth. To actually learn about these animals, we have to go out and capture them. And so I use live Sherman traps to set up trap grids at each of my localities, baited with peanut butter, oats, or what have you. And these are non-lethal little box traps. The animal goes in, attracts the bait, a door closed behind it, and then I can go extract the animal from the trap and then do my measurements and tag and everything. All of these animals I've been permanently tagging with RFID pit tags, so I can identify them from year to year for another project looking at year-to-year survival and population dynamics. Once the animals have been tagged, I can then measure and weigh them. And we also take some non-invasive samples, including those fecal samples that are really important for understanding that hormonal variation we were discussing earlier. Once the animal's been tagged and measured and weighed and everything, we can then send it on its merry way back home. But we'll then keep trapping that same location in hopes of catching the individual again, so we can then run the behavioral experiments to assess those questions of exploration and boldness. So pictured here along with me are a bunch of wonderful Berkeley undergrads who've been crucial in helping me get this work done over the past few years. This would not be possible without them, and I'll be shouting them out throughout this whole presentation and at the end as well. So once we have an animal captured and tagged, or we capture an animal and it's been tagged, it's an animal we know, an animal we've already measured, we can be like, oh, cool, okay, cool. We can now do our behavioral assays. How do we do that? And so we use this apparatus I have pictured to the right. We call an arena, and we use this arena for what are called open field tests, which are standard kind of measures of baseline behavior in individuals. Basically, all the open field test is you have an open neutral space, you allow the animal to enter the space on its own accord, and then you film its behavior over a standardized period of time, then code those behaviors in the software later to then generate behavioral data and compare between populations of interest. So what do these animals look like when they're in the arena? Good question. So this is Tamias speciosus that's about to enter the arena. This is a lodgepole chipmunk. And as you can see, it shot out like a rocket. And as you probably guess, its main goal is finding a way out. And it's very active and jumping a lot. And you might say, that makes sense. I imagine that's what they all do when they come out. And a lot of them do behave this way, but you still have variation. So this is a Tamias alpinus. This is the alpine chipmunk under the same experimental conditions, same arena setup. So it also shoots out like a rocket, but once it's out, it's a lot less active and a lot less, it barely jumps, if at all, and it's moving a lot slower. And so, I'm not a chipmunk. I can't read a chipmunk's mind. but being able to collect as much information about these responses as possible and get like a really fine-tuned understanding of what is actually what's they're doing, not if even if we don't completely understand why they're doing what they're doing, we can then at least make comparisons between the species and start coming to at least some preliminary conclusions. So once we have these videos, I then have to sit at a computer, myself and some other students. One of my students, Ketki, helped me a lot with a lot of this data, and made what would have been a 6-month process a 3-month process. So thank you, Ketki. So once you have the video, you have to sit down in a video software program, code all of these behaviors, and that's the actual data you use. The first step to that is you have to actually catalog all the behaviors that you want to measure and count in your video. And so that can be anything you observe and see, things like jumping, locomotion, grooming, etc. And so a few of the behaviors that I focused on was time to enter the arena, how long it took for an individual once the door was open to enter the arena on its own, time spent on the center of the arena versus time spent on the edge of the arena, which is a measure of exploration, basically how much of the arena space did it engage with during its time in the experiment, and how active it was, how much time it spent moving versus standing still. And we use those behaviors those measures, that we extract from videos as proxies for the more biologically relevant behaviors that we're interested in, things like boldness, activity, and exploration. And so after you sat at your computer for hours coding all these behaviors, clicking like, oh, that's a jump, up, it moved to the center, up, it moved to the edge, up, it's moving, up, it stopped moving. Once you've done that thousands of times for all of your videos, you can then extract that data, plug it into your statistical software of choice, mine being R, and then you can sort of see if there's any patterns between our two species in these behaviors, in our behaviors that we're interested in. And so here's some preliminary results from some of these analyses. I'm still pouring through some of the videos, but here's some of the results we have so far. Oh, and I forgot this was here, but just to kind of reorient you to the big picture question, right? So we expect Tamia speciosus individuals, the lodgepole chipmunk individuals, to be more bold and more exploratory than the alpinus individuals. And the boldness being how long it takes for them to enter the arena when they're first presented with it. Exploration being time spent on the edge of the arena versus time spent at the center of the arena. Basically how evenly proportionate are they engaging with the arena space. And then we also, the other question is individual variation. I won't talk as much about individual variation since this data set was being built, but I can talk about the first prediction. So first of all, boldness, latency to enter the arena. And so you can see here that alpine chipmunks take a lot longer to enter the arena than lodgepole chipmunks. And this is not only is this difference easy to eyeball from the means, but it's also a statistically significant difference. And so that's cool to see. Our more easily stressed out chipmunk is less keen on going into the big scary new box than our less stressed out chipmunk, our chill chipmunk. And so that's a cool result to see, because it makes intuitive sense, right? That if you are an easily stressed out animal, you're probably not going to want to engage with the new scary place if you don't have to. In terms of activity, general locomotion, again, this is just looking at how active individuals were, or what proportion of time they spent active in the arena. We see that the lodgepoles tend to be more active than the alpines once they're into the arena. And that tracks with previous studies that my lab and others have done on activity. Our lab in this species in particular, we've seen that lodgepoles, even in the fields, tend to be more active than alpines as well. And then, but what about exploration? What about how much of this new environment they're sampling? How engaged they are with this new habitat? Those results are a lot less clear cut. We don't really get a clear difference between each of the species. Again, this data set's still building. Maybe with further, maybe once we add more individuals in, this will change. But I don't expect this changing too much. And so we see that difference in boldness. We see that our more easily stressed out species, the alpine chipmunks, don't aren't as gung ho to engage with the unfamiliar. But we don't really see that when it comes to exploration. When it comes to, okay, once you're there, do you engage with that new space differently? No, we don't really see that. And so that's an interesting result to see, and not quite what we expected, especially seeing as boldness seemed to differ so sharply. And so that leaves us with some further questions to take away. So speciosis individuals, they tend to be bolder and somewhat more active, but we don't see a real difference in exploration. One of the reasons this might be is that there's a trade-off with exploration, right? So being able to explore your environment, being more actively engaged in exploring your environment, does give you more access and more opportunities to capitalize and discover new resources, but it also puts you at greater risk. For a heavily predated species like chipmunks, risk is no small consideration when you're engaging with the unfamiliar. And so, and it's often shown in the literature that there's often kind of there's a trade-off in exploration, that individuals that are more exploratory can often be at higher predation risk and actually less fit than individuals who are less exploratory. But then that also leads us to think how we actually measure exploration effectively. Is exploration just kind of a binned behavioral phenotype, whether you're exploratory or not exploratory? Are there different ways to be exploratory? And that's kind of where I'm thinking about heading next, and sort of thinking about exploration is, what are the different ways to be exploratory in the environment? What are the different ways to engage with and sample a novel environment? And how might that reflect in individual behavior and ecology? And then also going forward, looking at the repeatability of these behaviors as well. Kind of one of by the nature of this work, with catching individuals at free living individuals sort of at their whims, if they decide to go into traps or not, it's really hard to get multiple trials of individuals in the arena setup to look at how repeatable these behaviors are over time. And that's really crucial to understanding the role of behavior in an ecological or biological context. Sometimes exploration can be very repeatable. Sometimes it isn't. Sometimes they habituate to a particular assay, and then you have to switch up the assay. And so that's another sort of future direction that I'm looking towards for the chipmunks as well. So that's all I've got for now. I before we go though, I want to make sure to give a really special shout out to all of my undergraduates and field techs who've come out with me the past couple of years to help catch chipmunks, to help wrangle chipmunks, to help hike up and down mountains and set up these arena assays and do filming, collect samples, and all manner of thing to help make this project run. I could not do any of this on my own, and they are amazing, all of them. A lot of these students have gone on to grad school. Keki and Rochelle in the middle are both starting their PhD programs. Amy on the left, throwing up the peace sign, just started vet school. Brianna on the bottom left is a museum curator. Allie, who's on the very bottom right in the bucket hat, she just started a PhD program in Austin. Olivia in the pink in the top right, she just started a master's program. Melissa's working up at the Portland Zoo in the blue hat at the bottom right, and then Lark in the beige hat on the bottom, and the beige baseball cap on the bottom right, is working at a museum as a museum preparator at UC Berkeley. So I couldn't be more proud of them. And again, I just wanted to shout them all out for all their hard work. And of course, got to thank all of the sort of behind the scenes logistical support and all the people who funded me throughout this project. Again, well, the students couldn't happen without the students, but it also couldn't happen without money. So thanks again. And thank you all for listening so much. And I would love to take your questions.
can help facilitate those questions. So folks, if you put those in the chat or the Q&A box, I have a couple of questions myself to get us started. So I'm curious about chipmunk lifespan, and if you know, like, how long on average these two species live, and if it's similar, because I have a guess of which one has a shorter lifespan.
That's a really good question, Nora. In general, chipmunks can live about 3 years in the wild. So usually less. That's kind of what happens with small mammals, right? They're not known for longevity. And my hunch, and anecdotally, I think they can live longer in captivity. Like, I know people with like pet chipmunks back east, they can live 5+ years. And so in terms of differences between these two species, I don't actually know. We don't, that's kind of one of the things I'm trying to get at with my mark recapture work that I'm doing. I don't have that data yet, but hopefully soon I'll have a better answer for you to see if there's differences in mortality rate between the two species. My hunch being the lodgepoles have the higher mortality rate, but then there's the other side of that equation with population dynamics, right, which is fecundity. And so, it depending on how those two numbers balance each other out, you could still have lodgepoles could still have better population growth even with a higher average mortality rate if they tend to be more fecund or have higher like natal survival than the alpines. And so hopefully I can untangle a little bit of that soon.
Awesome. That's so cool. I was also like, oh, how cool would it be to be able to measure like the ratio of like the species ratio of what gets predated more. Also, like you said, if the population sizes are different, but like there's so many science projects that I wish could happen somehow, but seems impossible to know, like, which one gets eaten more proportionally. We do have a question in the chat, or at least a comment. Having pet rodents, I noticed they always like to follow walls. I wondered whether the walls gave them the means to remember where they were going, or if open space felt too exposed. In other words, maybe there's more than exploration going on.
Yeah, that's an excellent point, Linda. And that's something I definitely think of. And I definitely seems that like the walls, edges off, they definitely seem to treat it as refuge when they're going into the arena. And so it's still interesting to look at that proportionality of how long they're spending on the edge versus the center, because you think then the center becomes a risk-taking behavior, right? They're like, oh, the walls are security, going into the center becomes a risky behavior. And then seeing how that differs between our two, that's still something I'm pretty interested in. But yeah, I think there's definitely a broader dynamic with like risk and safety as well is definitely a part of it.
We have a question. How often are you trapping the same animal year after year, or even like month after month? Also, do these chipmunks tend to generally stay in the same area year over year?
Yes, good questions all around on that front. So in general, I'm still crunching the numbers for like specific rates of recapture rates and mortality rates, but just from my experience, I very rarely catch individuals from year to year. Again, chipmunks are small snacks. It's very rare that one lives that long. So it's very rarely I'll get an individual from year to year, and it's very rare that I've got an individual multiple years in a row, or more than 2 summers in a row. That's usually pretty rare. Usually I'll get a baby. Usually I'll get the baby in its first year, and then usually by then it's gone. In terms of within a trapping season or within the year, it varies. Some individuals I can catch repeatedly, very reliably. Some individuals I'll catch once and I never see again. So there's also variation in that as well. And then I think the last question was how do they stick to the same space? Am I remember that correctly?
Yeah.
So really good question. Chipmunks, there is some dispersal, but they don't tend to disperse as far as some other species. Chipmunk's home range is pretty small as far as animals go. It's only about a hectare. It's only about like a chipmunk that doesn't disperse will probably spend its entire life within the area of a soccer field, which for chipmunks, a lot of space, but still not a lot of space in the grand scheme of things, obviously. There is some dispersal that happens, and sometimes individuals will go a little further afield, but usually those chipmunks don't survive dispersal. And that's a common trend we see um, in rodents in general, is that dispersal usually means death, because just leaving an environment that you know and going across into familiar habitat just increases your risk exponentially. And so yeah, so most chipmunks will stay pretty close to where they were born, maybe disperse like a few kilometers away, but not that far.
Wow. I'm thinking now about the size of a soccer field and how many chipmunks I see. Do they have territories, or are they just kind of like evenly dispersed, or not evenly, but like randomly dispersed within that area? Are they fighting with each other for the best rock?
Yeah, so they're not territorial in the traditional sense that each chipmunk has like its own like little slice of area. What often happens is they're interspersed throughout the environment, and they kind of like will come into contact, and that's sort of how they sort of enforce it, but it's fuzzy that way, you know, so there's not like individual boundaries. It's more like if they run into somebody, they'll chase them off, or they'll have a little aggressive interaction. And so, but yeah, I think in terms of specific like burrows and stuff like that, they'll often guard those pretty jealously. Although there is some evidence that there might be some communal burrowing during the hibernation season. I don't know about my species in particular. As you can imagine, studying hibernating mammals is very hard. So it's not a lot of data on how they hibernate in the wild. But in other ground squirrels that are even species that aren't particularly social, they sometimes will tolerate each other to group together during hibernation just for the extra warmth.
Yeah. Oh, little cuddle puddle of chipmunks out of the snow. So cute.
Yeah.
Another question, Roy asked, what are the primary predators of chipmunks? And what is the most attractive bait to the two species that you're studying?
Yeah, great questions, Roy. So, excuse me. Get some water real quick.
Good reminder. Everybody hydrate.
I'm talking a lot. But the primary predators of chipmunks you're going to see, especially at this high elevation, like we're mostly above 9,000 feet. Mostly going to be raptors. So lots of your birds of prey, your hawks and things like that. This is too high up for most snakes. You're not really going to get snakes this high up. And then the other main predators you'll get maybe bobcats, maybe foxes, and then martens as well. We'll go after them. Probably martens are the most effective because they can chase them up trees and into little crevices that most of the other predators can't. But martens are fairly rare in the landscape, so it's not something chipmunks are having to worry about all the time. The predator they're probably most vigilant for are raptors, because those are the one they're most likely to run into. In terms of the most attractive bait, definitely peanut butter. They love peanut butter. They go crazy for it. I waffle between using peanut butter though, because every other ground squirrel in the Sierras loves peanut butter too. And so depending on the site, I can get, instead of getting chipmunks in my traps, I will get a bunch of Belding's ground squirrels or a bunch of golden mantled ground squirrels, or even like a chicory or all sorts of things. And so I find if I use like an oats or like a seed type bait, that tends to exclude the bigger animals and only the chipmunks really go for it. So I kind of have to balance that out. But if I'm at a site and I'm having no luck, I'll be like, okay, I'm going to bust out the peanut butter. We'll see what happens.
Awesome. Roy also asks, what is the likelihood of the chipmunks either biting you, researchers, or carrying disease?
Very good question. The likelihood of them biting is very high. They're ferocious little animals. Like I often think that If a chipmunk were scaled up, like it'd probably be the most scariest animal on the planet. Like they're just
T-Rex
so
for the Sierra.
even for the Sierra, maybe I could think.
T-Rex.
I think a big chipmunk could give a mountain lion a rest.
Yeah.
That's my personal opinion. But yes, they're very bitey. I've been bitten a ton of times. All of my students have been bitten, and it kind of comes with it just comes with the territory of handling them. In terms of diseases, most the diseases you have to worry about don't actually come from their bites. So the few like rodent diseases out there, so there's hantavirus. That's more of a concern with deer mice, with like Paramuscus deer mice, and it's a big concern in areas where deer mice are nesting. So not necessarily out in the landscape, but it's kind of where they're nesting. A lot of their feces have accumulated and dried and aerosolized, and then you inhale it, and that's how you get hantavirus, which is a really nasty virus that you don't want. But you're not really going to get that from a chipmunk bite, and you're not really going to get much of anything from a chipmunk bite. Rodents in general don't tend to carry rabies. And so the main disease you have to worry about from chipmunks and ground squirrels is plague. And so plague, the bubonic plague, is pretty, it's circulating pretty freely amongst in like all over, but particularly in the Sierras. But you get that from the fleas that live on the squirrels, not from the squirrels themselves. And so when we're out there, we always are wearing long sleeves. We handle them with gloves, and we always do very thorough little flea checks whenever we're working. And yeah, I've no one I've worked with has gotten sick, and no one I know who's worked with these animals has come down with plague. But it's definitely a risk that comes with it. And so, kind of my rule for the students is that so early plague symptoms are like pretty kind of run-of-the-mill flu symptoms, and so they're kind of pretty easy to ignore. That's kind of plague is very treatable. You can just use antibiotics, but a lot of people ignore it because they're like, oh, I just feel like kind of fluey. Who cares? But then it gets bad, and so I always tell my students like, even if you feel like you're remotely sick, tell me so we can go to the hospital, get you checked out. That's less of a hard sell nowadays post COVID. Like I think if even out in the field, if anyone starts to get like even a slight cough, they're like, oh my god, I have COVID, and like so people, I think students are a lot more aware of their health in that way than they would have been pre-COVID. So that hasn't really been as much of an issue as it may have been in the past. But yeah, so those are kind of the only thing you really need to worry about from the chipmunks. Otherwise, yeah, they bite, but they're small, so they can't do a ton of damage. And but still hurts though.
Ouch.
Yeah.
Some of my coworkers who worked on the Farallon Islands with cormorants would duct tape their sleeves so they didn't get bird mites up their sleeve. And I wonder if that would be helpful or useful.
That's not a bad idea.
It's like uncomfortable to have your sleeve.
Definitely uncomfortable. I know one student, she had like kind of little elastics that she'd put around her sleeves, just for like, and then like she would wear gaiters for ticks because she'd done a lot of like coastal field work, so she was really tuned to ticks. So she just wore gaiters all the time, and then I think wore like little elastic straps around her sleeves to keep it in. So yeah, it was smart.
Yeah, all the techniques. I have one last question. This gives you all a couple more minutes to think if you have any more, but the beginning you said the chipmunks are hard to ID in the field and tell apart. And I was wondering if you could tell us, or if you have any kind of general advice about not necessarily like how to tell an alpine from a lodgepole, but when you're trying to identify chipmunks, like what are the field marks or the parts of their bodies, their ears, their stripes that you really want to focus on that can help you tell them apart or ID them?
Excellent question. Yeah, chipmunks are hard to tell apart. And so some of the easiest things you want to start with looking is, again, the striping pattern. So the striping pattern can vary. So whether or not they have sometimes they'll vary between having 3 dorsal stripes and 5 dorsal stripes. And so that's usually a pretty good tell. A lot of the species in the area, there will be maybe one species with 3 dorsal stripes, one species with 5 dorsal stripes. So that's a good place to start if you're able to, if it's able to stay still enough. That's kind of the main trouble with
Yeah.
IDing chipmunks is they just don't stay still long enough for you to get a good look at them. Um, and so that just makes it really difficult. So the dorsal stripes are a good one. Relative coloration, there once you've seen a lot of chipmunks, there's actually can be a lot of color variation. So just as an example, the alpines tend to be a lot grayer than the lodgepoles. The lodgepoles have this kind of richer, sort of like reddish brown coloration, like especially around their chest and shoulders. While the alpines are kind of a more uniform, grayish tan all over. Um, and so that's and so when you have them side by side, it's pretty striking the difference. I think usually what makes it difficult is when you're kind of looking at them in isolation. Um, and so, but yeah, so striping, coloration, and then for coloration, you want to look at like certain regions. So like they might a lot of times they'll have like a certain coloration around their like chest and shoulders, and a different coloration around their hind legs and things like that. Oftentimes it's like browner here and grades into gray towards the hind legs, or they're brown all over. You can also look at sort of striping on the ears. So a lot of them will have like little white spots on the back of their ears, and some don't. And so that can help you out. And then also look at the tail. Some will have like more pronounced black tips on the tail. Some will have more uniformly colored tails. And so chipmunks are definitely something where it's like you have to see a lot of them, and you have to take a lot of different characters into account. There's often not going to be one thing. There's some species where there are. So there, once you leave the Eastern Sierra, some of the other species are a bit more distinctive. But a lot of the Eastern Sierra species really kind of grade into each other. And so it's a pretty fun exercise to learn them apart.
Awesome. Fantastic. Okay, last question.
Mhm.
What is your typical field season, the start, duration, etc.? And does it depend on the snow year for access to the site?
Yeah, so my typical field season's usually in the sort of early midsummer, usually starting in July, going into August. Um, yes, it does depend on the snow year for access to my sites. So a couple of my sites are pretty high up in the Hoover Wilderness area, and so on a good snow year, or on like a very heavy snow year, those trails can still be snowed out well into July. And so getting into those sites can be trouble. Although hasn't really been the case the past couple years. Bittersweet. I can get to my chipmunks, but I'm like, I wish there were more snow here.
We need
Um,
more snow.
yeah, definitely. Um, but yeah, so I usually start in July, and then I'll usually work until August. And I usually only spend about 5 days or so trapping a site. Usually by that time, I'm going to catch all that I'm going to catch. Um, it's usually I'll just stop getting new animals. Like maybe I'll get one or two, but usually by that point, you've caught everything in the area that's going to go into a trap. So you move on from there. Yeah.
Great. Well, thank you so much. That was really, really great. And I'm excited to for you to share some of this in person with folks who are going to be here next week. And I'm also so glad that all of our virtual attendees can learn from you as well. So we just really appreciate it, and just fantastic. I love chipmunks. There's so much to learn about them. It's so exciting that you all in your lab and have been doing this great research, and that there'll be more to come. So thank you so much.